Cancer Therapeutics and Delivery Systems. My name is Tim Shannon. I'm a partner at Canaan Partners, a venture capital firm that's active in the oncology space and the biotech space in general. And I'd like to thank you all for joining us today in what we hope will be a really engaging 90 minutes. I think for those of us who've been involved in this area, there's nothing more exciting than to develop a novel cancer drug and see it benefit a patient. It's incredibly rewarding. I think in today's session, as some of our speakers present their work, you'll see how impactful Yale's been in this sphere, and particularly the Yale Cancer Center, and really helping to herald the great progress that we've all seen in cancer therapeutics in the last decade. But I think Yale and the Yale Cancer Center recognize that this is really a community effort and requires extensive collaboration and networks to make this all work from start to finish. That involves universities, governments, industry, as well as investors, all playing a role in this very important activity. So really the goal of today's session is really twofold. It's for Yale really to continue to broaden that network and that community with whom they engage, again, to try to live out their passions of bringing promising and novel therapeutics to patients. But it's also importantly for you in the audience, to learn about what Yale has to offer and how you might be able to become part of this community around the Yale Cancer Center and share that mission and that passion to do great things. So some of you may have already been involved in the Cancer Center. Some of you, this may be your first opportunity, but for both of those groups, we certainly hope it's not the last opportunity. And this really catalyzes more engagement with the Cancer Center. Just in terms of some housekeeping issues, the audience in general will be muted, so you don't have to worry about the noise you're making in the background. But we really do want this to be interactive, and the chat room will be live, and that will be the vehicle with which we'd like you to raise questions during the symposium, and particularly for panelists, after you hear what panelists have to say, and your questions can pertain to their comments, or your questions can be really as broad as you like. So again, we would really encourage participation via the chat room. That will be open, and we will be monitoring this. Finally, this webinar will be recorded, and I think I just saw a chat room question come up in that regard. So the webinar is being recorded and will be available after the session. So with that introduction, I'd like to welcome our first speaker, Dr. Charlie Fuchs. Dr. Fuchs is the Richard and Jonathan Sackler Professor of Medical Oncology and Professor of Chronic Disease Epidemiology at Yale. He also is the Director of the Yale Cancer Center and Physician-in-Chief of the Smilow Cancer Hospital. Charlie, thanks very much.
Tim, thank you. And I want to welcome all of you to our second in this series, Yale Engage Cancer, which really has been an exciting opportunity for us to reach out to a broad audience. And I want to just say that, we're obviously engaged now in a global pandemic, which I think has to some extent been the forefront of our attention in healthcare and drug development. But, as we get past this, and we're all looking forward to that point, we are reminded of the fact that in the 21st century, cancer is really the great challenge that faces American medicine. It is the focus of so much work clinically in terms of support from government and philanthropy, and really the focus of investment in drug development. And I think one thing we all share, we're a diverse group on this call, from academia, pharma, biotech, the investment community. We're diverse, but I think what we all share in common is a commitment to innovating in science and taking those discoveries and moving them into meaningful improvements in the treatment, early detection, and prevention of cancer. You know, I'm pleased as the director of the Yale Cancer Center to be at a place with an extraordinary legacy of cancer research really over the century in cell biology, genetics, chemical biology, pharmacology, immunobiology, among others, and clinical research. And, you know, as the NCI Comprehensive Cancer Center for Connecticut and New England, we carry that responsibility with great pride. Beyond the great science, we are really proud of our clinical programs. In fact, the building I sit in now is Smilow Cancer Hospital, which is celebrating its 10th anniversary as a center now that provides care to 48% of all newly diagnosed cancer patients in the state of Connecticut and patients well beyond that region. And with that has really been the laboratory of moving discovery in the laboratory into meaningful clinical treatments for cancer. In fact, one opportunity of great pride I wanted to share is that this year, 4 Yale-led clinical trials will have led to new approvals in the oncology space with the FDA. So not only are we committed to great science, but we're committed to moving that science into the clinic and into ultimate approval. Tim has assembled for today's forum just a panel of rock stars who not only do great science, but actually are committed to moving that science into the clinic through their own work and through partnerships, by starting companies, by working with pharma and biotech. And I think each of their stories are really going to be interesting for us. I also want to particularly thank Josh Bilenker, who's the Chief Executive Officer for Loxo Oncology, for joining our panel, which I think really adds more to the richness of discussion of how you ultimately move drug development from science into ultimate care of patients. You know, one thing we want to do in these forums, and I say this is the second one, the first one was on immuno-oncology and very well received, is in this forum focused on a key aspect of cancer treatment and research, is really that it be conversational. We want interaction, not only between our panelists, but with all of you to answer your questions. And more importantly, we want this to be the beginning of a conversation. We hope that this stimulates new partnerships between our colleagues in Yale and you, because ultimately, as Tim indicated, really to make a difference in cancer treatment and cancer research, we have to be working together. So, you know, please feel free to reach out to me, to the panelists, my colleagues, both during this meeting and after. We'll remind you of that because we want to work together in this fight against cancer. So Tim, thank you for leading this, and I look forward to an exciting forum.
Thanks very much, Charlie. So the slide you're seeing now reflects, the agenda for today. So the way the session will be set up is we'll have brief 5-minute presentations by the presenters you see listed on the slide, followed by a large period of time to enable discussion and question and answer sessions with panelists. And again, we really encourage your participation here. Our goal here is really to provide information that you're looking for. So your questions are really what we need to help deliver that to you. In their presentations, each of the speakers will talk a little bit about what their core scientific expertise is, and particularly what sort of questions they're trying to answer, with the goal, again, of ultimately impacting cancer care. In their presentations and in part of their comments, again, they'll also talk about some of the ways they have collaborated, to accomplish what they've accomplished thus far, but also, set up the stage for additional ideas about collaboration with the broader community. So the chat room will be open throughout. Please, again, feel free to put your questions into the chat room, and we'll be monitoring those and bring those forward. And again, all speakers, we'll start with about a 5-minute presentation before we move into discussion and the question and answer session. So with that, I'd like to welcome our first presenter, Ranjit Bindra. Ranjit?
Great. Thanks so much for having me today. And thanks everyone for joining. I'm a professor of therapeutic radiology, the Yale School of Medicine. I'm also co-director of Yale's Brain Tumor Center. We'll go on to the next slide. So one thing I'd like to start with, as Dr. Fuchs mentioned, is we have quite a presence along the translational research spectrum at Yale. And I've been here both as a medical student and as a ninth year into faculty, and it's just a wonderful place to be. On one end, we have disruptive cutting edge science, and just a few examples that you won't hear about today, but I wanted just to highlight Jason Crawford and Seth Herzon finding gut bacteria that produce entirely new DNA damaging agents. In the middle there, Mark Saltzman, Don Engelman, and Peter Glazer with nanoparticles and tumor targeting peptides. At the bottom there, we have from Aaron Ring's lab, an entirely new secreted immune checkpoint. But on the other side of the spectrum, we're known for pivotal studies and excellence in clinical research. Here's a study, two studies shown by Dan Petrolak and Roy Herbst, and also work from Pat LaRusso, really defining high-level clinical trial designs. And behind and in parallel to all that, we have a very vibrant and active biotech and pharma community, and it's really driven by the heart of it at the Yale Office of Collaborative Research. And certainly you can check out their website, and you can see there just some logos of the companies that have been recently started, and notable that some of these companies are associated with the papers that I just showed you earlier. And we'll be talking about some of this work throughout the session today. Now we'll move on to the next slide. So just a little bit of background about me. So I sort of also live in multiple nodes of that research translational spectrum. I'm a PI of an NIH-funded DNA repair lab, and our lab is mainly focused on oncology, synthetic lethal drug screening, with a focus on DNA repair and metabolism. And we've been fortunate to publish a number of high-impact stories recently in some of those journals shown there. I'm also a radiation oncologist, and as a co-director of the Brain Tumor Center, I'm involved in a lot of glioma clinical trials. These are some of the cooperative groups that we work with here at Yale. And then sort of on the evenings and weekends, I'm very much involved in the biotech world and an active biotech entrepreneur, most notably a company you may be aware of, Cybrexa Therapeutics, which will be in the clinic early next year. We'll go to the next slide. So I just wanted to give you really just two case studies of some of the work that we are doing and how we've been able to translate that from the bench into the bedside and really into biotech. This is a story that we recently published in Science Translational Medicine, and then more recently in Nature and Nature Genetics, where we found that oncometabolites like IDH1 mutations that induce 2-HG unexpectedly cause an HR defect or a BRCAness defect that causes PARP inhibitor sensitivity. And we made that discovery just a few years ago, and it's really a collaborative effort across multiple labs here at Yale, including Peter Glazer, Murat Ganel, and Stephanie Helene. But we've been fortunate to be able to rapidly translate that into the clinic. And these are some of the trials that are shown here. But most importantly, in the bottom right, those are the clinical trials at Yale that really made these clinical protocols possible. And we're now taking specimens from these trials and bringing them back into the lab to try to understand who responds and who doesn't, and what's the molecular basis for that response patterns. And we'll move on to the next slide. And just a second, more recent story that we published in Nature Communications on the metabolic front. We found that a phosphatase, which is upregulated or mutated in a number of cancers, including brain tumors, actually has an epigenetic silencing function that had not been described previously. And this leads to silencing of a key NAD metabolism gene called NAPER, which essentially inactivates the press handler salvage pathway shown there and leads to exquisite sensitivity to a class of drugs called NAMT inhibitors. Now, these drugs have actually been tested extensively in clinical trials, and many of them have been shelved or stymied because of lack of good biomarkers. And we actually believe we have those biomarkers now. And again, leveraging the Yale OCR and the Blavatnik Fund, which we can get to later, and Connecticut Innovations, a local state-backed VC, and the PITCH program developed by Craig Cruz, we've been able to start a company to develop a new class of NAMT inhibitors, and we hope to bring those into clinical trials soon. So with that, that's just a flavor of what we do, and really looking forward to the discussion today, and thank you.
Thanks, Ranjit. Much appreciated. We'll save again collect questions throughout the presentation. So now we'd like to move on to the presentation of Sidi Chen. Sidi?
Sidi, please turn on your video if you're able.
Sidi, can you unmute and open your video?
Tim, maybe we could advance to Craig's slides and give Sidi a chance, and we can come back to Sidi in a moment.
Sounds like a good idea. So again, we'll come back to Sidi Chen, but let's move on to Craig Cruz while we try to work out Sidi's kinks.
Great. By the way, here we go. Thanks, Tim. So I am the John C. Malone Professor of MCDB and Joint Appointment in Pharmacology, as well as in chemistry. And I'll be talking a bit about my role in running the Yale Center for Molecular Discovery, which is the small molecule and RNAi screening center that is here at Yale. Next slide, please. So I've been at Yale for 25 years, working at the interface of chemistry and biology. When I started here, it was primarily focused on mode of action studies of natural products. And this is one molecule that came from those efforts. It is a derivative of a microbial organism, derived organism, compound that is an anti-tumor compound that is a proteasome inhibitor. And so my lab identified the mode of action of this molecule, did some medicinal chemistry. I founded my first company, Proteolix, 17 years ago, that turned this Yale finding into a drug candidate. And this was approved for relapsed refractory multiple myeloma in 2012. Proteolix was purchased by Onyx and ultimately by Amgen. And Kyprolis now does over $1 billion in sales for, again, refractory relapsed multiple myeloma. But since then, next slide, I've been working on the flip side of that, not blocking degradation, but actually inducing degradation. And so my lab's played a key role in developing this new field of targeted protein degradation, where we can, through induced proximity, drag proteins to the quality control machinery of the cell, inducing its degradation. And I've been working on it for 20 years now, but a key part of this transition in terms of translational impact was the founding of Arvinas here in New Haven in 2013. This is from their IPO in 2018. They now have 2 clinical candidates in trials, one for breast and one for prostate. And moreover, at least for their leading one, the prostate, they've been able to demonstrate that this technology works in humans with respect to actually targeting and decreasing surrogate markers for prostate. And so that was quite exciting. And I continue to play a role in advising Arvinas. But I wanted to spend just a little bit of time talking about another hat that I wear on the next slide, and that is the Yale Center for Molecular Discovery. Founded 17 years ago, and this is really the nexus of translational research at Yale with respect to developing assays, screening compounds. You can go on to the next slide. And so we have the resources where we have small molecule compounds, we have siRNA collections. You can go back a slide. Now forward. And so we have a team of professionals that are helping the Yale researchers to translate their academic findings into an assay, and then screening small molecules, SI, using robotics, and then helping to interpret those assays and those results with the whole point of coming up with impact. And that is defined by papers, that's defined by grants, but it's also defined by starting companies. And so where we stand now is that we have the infrastructure, we have the network, we have the resources, but we want to reach out to establish more industrial partners to help guide us, help prioritize, help help direct where we should be focusing our efforts. What are the unmet needs in industry? What are the new indications that we're not aware of? And so we're really like to serve as a bridge, if you will, between the diverse collection of cancer researchers at Yale and the greater world. And so with that, I'll stop and turn it back to Tim.
Thanks, Craig. So I think we have Sidi Chen's logistics worked out, and we'll go back to Sidi Chen's presentation. Sidi?
Yep. Can everyone hear me? Good. Okay, thanks. Next slide. So my lab's focus primarily is cancer immunotherapy, and we are trying to use multiple approach to improve cancer immunotherapy, which is currently a transformative therapeutic modality, but still having major setbacks by more than 70% of patients not responding. So the first program for us is to discover novel immune checkpoints or T-cell regulators that are important for the tumor microenvironment, and in order to overcome the current therapeutic resistance. And once those new molecules were discovered, we will be able to identify the therapeutic strategy for them. And then the second program is to engineer immune cells themselves, like CAR T cells, and develop a therapeutic strategy to infuse those cells back to patients and as a cell therapy modality. And the final one would be a program based on cell and gene therapy. And in this case, we call it MAEGI, which I'll elaborate later on. And next slide. So MAEGI is a new class of cancer immunotherapy, we believe, even though it's at early stage. It is coined as multiplex activation of endogenous genes as immunotherapy, MAEGI. So we think this is a new strategy as compared to all of the existing ones, because cancer cells, they have a lot of signals that are considered non-self, meaning the mutations that distinguish them from the normal cells. But in order to find and kill the cancer cells, we better off amplify such signals. And one of the most efficient way we figure out is by CRISPR activation. Just crank up all the non-self signal so the immune system can see the cancer cells clearly and then come to destroy the cancer cells and kick off the immune cycle. Next slide. And then we have done a few proof of principle experiments and show that these works quite well in preclinical models across multiple cancer types, including breast, melanoma, and pancreatic cancer types. And we hope to continue to translate these new therapy modalities and use them either alone or in combination with what are already available. Thanks.
Thank you, Sidi. Next, we'll go to Faye Rogers and listen to the presentation of her work. Faye?
Thank you, Tim. So I am an associate professor in the Department of Therapeutic Radiology, and my lab focuses on understanding DNA damage response. And so I'd like to tell you a little bit about how we've been using that interest to develop a novel form. Next slide, please. So oncogene amplification is considered to play major roles not only in the formation of cancers by amplifying genes that provide growth advantages such as increased cell proliferation, angiogenesis, and decreased apoptosis, but also in later progression to states of metastasis by amplifying genes that facilitate migration and invasion. So it's not a surprise that gene amplification has become an important therapeutic target in the development of precision medicines. And so the current drug development strategy has been to target the overexpressed protein products that are produced due to gene amplification, which has resulted in major drug breakthroughs such as Herceptin and other tyrosine kinase inhibitors. And so our lab, as I said to you before, is really interested in understanding DNA damage response. And we have taken an approach to manipulate the DNA damage response network in order to find a novel way to treat these types of cancers. So the cell has already in place DNA damage response mechanisms in order to preserve genomic integrity. And our goal is to hope to manipulate this network by forcing cancer cells to go towards the pathway on the right, following excessive DNA damage to activate apoptosis, as opposed to normal cells which can choose to repair DNA damage. And so by hijacking the cell's own machinery, our goal is to reduce the normal tissue toxicity that might be experienced by these kind of drugs and prevent off-target effects. So next slide, please. So our focus has been to directly convert the amplified DNA into DNA damage. And so by doing so, we have designed oligonucleotides that can bind to specific sequences within the amplified gene, which is then forms a triplex structure that is recognized by the cell as DNA damage, and through replication stress leads to excessive DNA damage, specifically at the amplified regions. This excessive DNA damage then can activate a DNA damage response as a result of all this damage, leading to the activation of a P53 independent pathway. And so we believe that this drug design platform can actually have far-reaching approaches due to the fact that there are over 500,000 unique triplex targeted sequences throughout the human genome. And in fact, there are more than 460 amplified genes across 14 different cancer subtypes. So we believe that we can specifically use this drug design platform to target many different types of cancers that are characterized by gene amplification. And thank you.
Thanks, Faye. Very interesting. Now we'll move on to the final speaker who's joined our panel today, and that's Josh Bilenker. Josh comes from outside of Yale, but he certainly is part of the community, living in Connecticut and starting his last company here in Connecticut. So we're thrilled to have him here. Josh can really speak to today's topic from a number of angles, given his background. So he's a trained medical oncologist. He's worked at the FDA. He's worked in venture capital. And then most recently, he's started his own, incredibly highly successful company, Loxo Oncology, which recently was acquired by Lilly. So now he sits inside Eli Lilly and is really one of the senior managers there in charge of Lilly's strategy in oncology. So again, we're really pleased to have Josh here today, and he'll just offer some comments on what he sees as the challenges and opportunities in developing novel cancer therapeutics and in regard to collaborations of companies and academia. Josh?
Thanks, Tim, for the kind introduction and having me here. I prepared like two sets of remarks, and I think one of them may be appropriate for this session. I might save the other, but I'll try to get them through quickly, Tim, and if you want me to elaborate on anything, please interrupt me or come back at the end. But one set of remarks I thought might be interesting and relevant to the attendees is the topic of what are the types of relationships an academic lab can have with industry, and what are the sort of risks, rewards, and implications of those relationships. And I really break them into sort of three buckets with sort of increasing levels of intensity. I think in the sort of lightest intensity relationship set is, I'll call it the advisor-expert model. Where the goal of the relationship with industry is really to raise awareness or to attract resources in a global sense to a problem set that you or your lab thinks is important to human disease. And you can have this influenced by, say, providing free advice even, or providing paid expert advice as a retained consultant, or maybe being a scientific advisory member to a company or a small company or a large company, or a guest lecturer in a large pharma setting is another example. And the output that you're expected to supply, whether there's sort of a compensation return or not, is basically your ideas. A lot of your remarks are going to likely refer to public domain information, but it's really your expertise. And again, the goal from both sides is just to bring awareness and education to perhaps a biology set of questions that needs your particular input. So that's level one. That's light. That's no strings. That's kind of easy to come and go with. And I suspect many of you have already engaged in that type of relationship. One level up from that is probably the meatiest, and I'll just call it, for lack of a better sort of bucket term, let's call it some kind of sponsored research agreement that covers you and your lab or your group, and probably involves your university signing off. And in this type of agreement, the expectation is that your lab or know-how is providing some kind of assay expertise, where there's a data readout expectation that is somewhat proprietary, between you and the industrial partner. And I've seen this transpire in both what I'll call an external model and an internal model. The external model, there's a transference of the assay to the partner. They run the assay. They perhaps share the results back with you, but your academic lab mission is not asked to be amended to supply the data. Ultimately, it's on the part of the industrial sponsor to do that, but you've enabled the tech transfer of that assay. And then on the internal relationship, you're really the one running the assay, and therefore you're kind of on the hook for deliverables, and you have to balance the academic mission with the industrial partnership and how to staff that and resource it. But either way, the goal here is to create data, not just your ideas. And those data, again, are internal or externally generated. And then the third level is probably the most intense. That's the company founder or co-founder role, where it might subsume the other two categories. There might be a sponsored research component, there might be an advice component, but really, you're really putting your kind of currency, your personal currency, behind a set of ideas. And there's some expectation that commitment, with all its conflicts, and my next set of remarks tries to handle some of those conflict issues, but really the behavior involves, your personal sort of stake in this thing. And you might be involved in mediating conversations with tech transfer or investors or entrepreneurs as they come around an idea, or pharma executives if it's a big pharma relationship or subsidiary. So I think that's the most intense, and I think it has its own trade-offs. But with that, I'll pause and see if, Tim, you'd like me to stop or elaborate.
Josh, I think you're on a run. Why don't you spend another minute or two elaborating, and then we'll then next look for some panel reaction to your comments. So continue on a little bit more.
All right. It might be too much to handle the complexities of, I'd like to, I have sort of a dedicated set of remarks about the frictions that I see as academics, really set up one of these three relationships, and how do they, I'm just being distracted by some inappropriate comments on my screen. Maybe I'll ask the moderator to look into that.
My apologies, we're taking care of that now.
Okay. I think all of these relationship sets involve some soul searching, which I think happens at two levels. It happens at the personal level, like, what am I comfortable with? How do I want to use my time? How do I feel comfortable or uncomfortable with the idea of industry being part of my academic mission. And then I think there's also a time management, sort of implication to some of this. How do I want to spend my time? Do I like the idea of being around a company? Do I like the idea of being in the halls of a large pharma periodically? So, I think it all looks like roses. I think the worst assumption set to go in with is like, oh, industry is this sort of undisciplined, unlimited source of capital that's just going to augment the academic mission of my lab. I think that's the most simplistic and, quite honestly, unhelpful for both parties mentality. And it's a very common mentality I encounter. I think it's sort of a dishonest partnership if that's the thesis, because it misunderstands what I think the true value of the partnership was intended to be. And so happy to unpack that a little more, but I want to say it out loud because if one listening to this had that expectation, I think it's really worth another set of discussions that maybe the panel wants to unpack.
Great, thanks, Josh. So that's a great entry into our panel discussion. So if we could just ask all the panelists to open up their mics and turn on their videos, and we'll move into the discussion session of the meeting. So again, a reminder to the audience, please bring forward any questions you want into the chat room. Again, we'd very much like to hear those. So let me maybe just start out by asking the panelists, just to, react a little bit to Josh's comments in terms of your experiences, in terms of interactions with investors or biotech or pharma. And maybe comment a little bit on how you have thought about that as you've sort of entered into those relationships, and how you've managed some of those issues and some of those tensions.
So I think that, Josh, you bring up a good point in terms of just expectations on the faculty's part, and in terms of what they hope to get out of these relationships. I'd like to think that that isn't that big of a problem these days. It has been a problem, in the past. But OCR and the entrepreneurial ecosystem at Yale does a really good job, I feel, in terms of educating faculty and helping them set expectations, figuring out what's possible, and so at least compared to when I started, again, 25 years ago, I find that the younger faculty that are now thinking about these types of partnerships are much more sophisticated in terms of what they need to prove with respect to what they bring to the table. And so I'm sure that there will always be exceptions, but I've been impressed.
Thanks, Craig. Anyone else? Ranjit, any comments? I know you've sort of been involved in some of these situations. So
yeah, it's a really great question, and I've been involved in a lot of different levels, including the companies that I've started, and also companies that have reached out to me. And there really is a sweet spot I found. I mean, I found that some smaller companies and larger companies really understand, as does the academic side, the balance and the symbiosis of what each party can offer. And if you can get it right, you can really, the academics can offer expertise or think about things the way the pharma or biotech side wouldn't necessarily, and really vice versa. So I think there's an art to it, and I think it's actually quite important to recognize really a lot of the comments that Josh said. So.
Good, thanks, Ranjit. Appreciate that. Anyone else with any comments or thoughts? I mean, I think it's important guidance, and really these interactions, like all interactions, require trust and familiarity, and really are based on relationships. And I would say part of the purpose of meetings like this, again, is to build those relationships, again, to sort of navigate those inherently or potentially tricky issues between an academic scientist and an investor or industry. Let me see, so we have some questions coming in, so let's move to some questions from the audience. The first one, I'll just read out. As an investor, I'm intrigued by the YMCD. Is this facility and the professionals working there accessible to people, startup companies outside of Yale?
Yeah, no, I can handle that. So the answer is yes. We have mechanisms in place that allow the screening technology, robotics, the compounds, the assay development at prices that are, of course, consistent with Yale policy. But yes, it is possible. In fact, several startups, Yale-based startups, use this as a way to augment in a virtual way some of their challenges of just getting off the ground. And so it really, I think, is a nice bridge again to translational work from Yale.
Thanks, Greg. Another question.
Can I interrupt 1 second? I want to apologize to the panel and the audience. There's obviously some individuals who are disruptive and posting totally inappropriate things. And I know that Kathy Lynch and her team are working to remove it. So I just want to apologize for what is a great forum, and I'm sorry to interrupt, but I just wanted to explain that.
Appreciate it, Charlie, and we will carry on for sure. And make sure we pick up the appropriate questions from the chat room. So one of these, Faye, was for you, and I don't know if you saw it, but I think, again, people find the basis of your approach and the potential scale of your approach like to be really interesting. So the question really was framed around potential downsides of this in regard to toxicities and how that might be able to be managed. So maybe could you just how you think about that in terms of managing the toxicities, and also maybe on delivery. So how would you think about delivering these therapeutics to target tissues?
So yes, I think the beauty of our approach is the limitation of having toxicities. So you might imagine if we designed an oligonucleotide to bind to a specific sequence within a gene, that in a normal cell, you would only have 1 or 2 copies, and I'll use HER2 as an example. A normal cell, you would only have 1 or 2 copies of the HER2 gene, meaning there would be only 2 binding events within that normal cell at the most. And that this kind of DNA damage would be effectively repaired by DNA damage response pathways that we know can recognize and repair this type of DNA damage. Conversely, in a cell that has gene amplification, there would be multiple binding events, the induction of excessive DNA damage that would then overwhelm the repair capacity. And so the cell would then choose as an alternative to activate apoptosis. So I think that is the beauty of our approach, because it takes advantage of the cell's genomic integrity and those pathways that are put in place to maintain that. So normal cells can repair, they will survive, and they will be fine, while the amplified cancer genes would have excessive DNA damage and they would choose to activate their own cell death. With regards to delivery, we have been working with Dr. Mark Salzman and have been able to use nanoparticles to effectively deliver these oligonucleotides specifically to tumors. And we've been very successful with this using the nanoparticles. We have very good uptake of our oligonucleotides to tumors.
Great, thanks very much, Faye. Appreciate that. Next question comes from the audience, I think is again, a bit of a follow-up on, the potential academia industry tensions. So, maybe the question would go out to each of the academic PIs. How do you manage, sort of your Yale responsibilities of training and developing students, PhDs, postdocs, with issues that pertain to your corporate relationships? Maybe, talk a little bit about how, you all just manage those issues within your lab in terms of who, as it might pertain to sort of who's involved in purely your academic work versus who might be involved in industry work. So let me just open that up for anyone.
Great. I'll jump in first. Actually, one thing that I've learned in the recent years is that a lot of trainees have different aspirations and different interests, and it's not you'll have graduate students and postdocs, especially in these times, that aren't may have an academic focus for their career, may have pharma, biotech, startup, consulting, venture capital. And I actually now often will, when I have career development discussions, I'll actually first ask my trainees, where do you want to be? And, there's no judgment about where, every part of this life science development process is very important. And it'll help me know what you want to do in your career, because that actually also helps me how much to, involve some of our trainees when appropriate, obviously, in some of the work that we do outside of Yale. I think that I'll leave it at that as a first comment, but let some other folks comment as well.
Anyone else want to add?
I can simply say that the way I enter these collaborations, partnerships, I always make it clear, and of course Yale requires that work that's done in my lab has to be publishable with, of course, the right reviews and checks and balances, but it has to be, in theory, publishable. But to Ranjit's point, I find that there's a wide spectrum of people wanting to sample industry. And in fact, I think that's a big knock against academia right now, is that we don't do enough to offer them real world opportunities. So I find that these relationships that I have with the private sector actually help my lab in terms of recruiting people that want to learn how industry works, and specifically, what type of skill sets they need to or can pick up while in my lab. And so I find that it is a really nice balance, but I haven't had a problem in terms of students. So maybe it's because there's a selection there that they come to my lab wanting specifically to gain that skill set.
Yeah, great, Craig. And I'll just add, because I'm, local here and involved a bit, to speak to that hunger and interest. We at Canaan, with both Yale and UConn, have started at Yale what's called the Yale Canaan Fellowship Program, which really, is open to MD or PhD students who are really interested in the more corporate side of this, be it in startup biotechs or on the investment side. And again, the interest in that program is exquisite, and the talent is frankly outrageous. So again, I think there certainly are large, segments of the new world of trainees who really are trying to learn this other side of the universe early on in their careers. And I think it's great, because I think the less this is a sort of a two-sided game, and the more it's just a continuum, I think the much more productive it will be, and it frankly needs to be a continuum, obviously with appropriate precautions to protect, academic integrity. But I think the world would be a far better place, the more seamless we can sort of make this for people. Josh, let me throw it back to you. I mean, you, as I've mentioned, have, a real wide aperture in terms of what you've gone, your work experiences and your successes. I'm just curious now, as you sit inside, one of the largest pharma companies, and you really have oversight in terms of, a lot of their strategic thinking around oncology. Maybe just share, with the panelists and as well as the audience, how Lilly's thinking about their oncology strategy, and what might be similar in regard to Lilly with other major companies, but also, what might be different, sort of with a focus on where you see the ball moving. And, back to sort of Craig's opening comments, really trying to give people an understanding of what the big oncology franchises are going to be looking for, 5 and 10 years down the road, when investigators' work, starts, coming online.
Yeah, I mean, big pharma environments are complicated places. I think they're confusing from the outside to try to interpret. They were confusing to me when I was working at a small company, and they must be confusing to many of you as sometime collaborators. And I think there's like two or three reasons for that. One reason is large pharmas are constructed in what they'll call, most of them, as a matrixed organization. So that means there are these technical disciplines that are somewhat independent of each other. So the commercial group is the commercial group. The biology discovery group is the biology discovery group. The pharmaceutics people are their own group. The business development are their own group. And they might report up into common structures, but if you as an outsider are interacting with somebody, it's really hard to, from their business card, sort of know what political entity they represent in the larger company. So you may think that you have the ear of a company or not, but it's really hard to know what political sort of dynamics are going on behind the scenes that may or may not allow that person to deliver on what you're talking about, or deliver in the way you'd hope. And so one of the things I've tried to do at Lilly is break that down as much as possible and make our portfolio as coherent as possible, all the way from target ID to first approval, and not have these gating, technical go no goes, where at any moment the rug could be pulled out. And that's the problem with the matrix model, is that they often can lead to incoherent decision making. I think a second, so I think two other themes worth mentioning is the commercial entities at these companies, the ones who are marketing approved products, have a lot of say on what happens in R&D. They have this idea of what moves the needle, and they still see the world through the lens of conventionally defined markets. Lung cancer is bigger than melanoma, and melanoma is bigger than neuroendocrine tumors, and neuroendocrine's bigger than some even rarer orphans. So they're going to see the world commercially, and like they'll use this like troubling phrase, what moves the needle? Can we make $1 billion on this drug a year? Can we make $5 billion a year on this drug? If it's just 200 million, we don't even want to hear about it. So there's this, in many large pharmas, this disproportionate commercial voice that affects strategic thinking, and that voice is not futuristic. It's backward looking at sort of what are the franchises that already exist and are proven, and can we bolt on to those existing franchises in ways that sort of feel familiar, so as to fill in holes from patent cliffs. And that's not, in my opinion, the right way to look toward the future. And then the third, I think in companies that have more R&D influence at the top, they are thinking about futuristic platforms. They are thinking about thematic bets that they feel they don't want to feel like they've missed out on. Things like gene therapy, cell therapy, RNAi, CRISPR, like big delivery, big idea, new modality kind of thinking. And there are a handful of companies that are willing to risk capital at just not fear of missing out. They're not going to, quote, overpay for that optionality, but in other words, in the absence of an approved product, but they'll place some bets. And I think that's sort of a third theme that I think can drive some collaboration.
So can I just ask a follow-up question, Josh? You say some companies have a greater appetite for that type of early platform, they don't want to miss out type of approach. What distinguishes those companies from their peers?
Some of it's corporate structure and the force of individual personality, which is the most surprising and least satisfying answer I can give you. So, you've been in a complicated room of decision makers. There are some rooms that are dominated by certain voices and that's separate from their title sometimes. And some others have org structures that represent that. Who's at the top? Is the CEO of the company, does he come out of commercial or does he come out of research, or she? It's a very, what is the last board meeting they had? And what pressures did the executive team feel coming out of that board meeting? Was the board feeling like cash poor and risk averse, or were they fear of missing out because their competitor just did a big deal that they feel embarrassed that they didn't get to look at? Like, these are these like incredibly soft, unknowable things that prevent me from really answering you in a coherent way.
But you know, you've been on the other side as CEO of a smaller company that was now you're on the other side. How can you, as a CEO of a biotech, position yourself so that you can take advantage of some of these unique characteristics of the marketplace? Can you create a market without losing the bird in the hand?
What do you mean by bird in the hand?
So when you want to sell yourself, right? If you get an overture, how do you make sure that your company is what is the market value of your company if you only have one interested party?
Well, if you look at like Form 14 SEC documents that describe the anatomy of most M&A deals, 70% of them are one-party deals. There's not a competitive process. So that's just how it goes the majority of the time. There's only one party at the table. The second assumption I always make is every company that's pre-commercial is for sale at all times. So like the notion that like there's this board of directors or investor base that's holding out, most credible offers are considered and usually taken. So I don't really see this sort of waiting for perfection mentality. Everybody's for sale. If you're a public company, your price is listed on the ticker every day. There's some takeover premium that needs to be added to that, whether that's a 20% premium or 100% premium is sort of up to the multi-party dynamic that you alluded to earlier, or the swagger of your banker and your CEO, or just the lack of discipline or intense discipline of the counterparty. I mean, these are incredibly like human one-off moments in time. Like there's, again, no strategic way that I can give you first order advice. That's just been Tim, what have you seen as a board member in this?
Yeah, no, I agree. I think, first of all, most M&As are one-party M&As, so the notion of this competitive, process is not the norm. It's usually the exception. But I do think most boards have to be open to respectable offers, because boards are representing the interest of shareholders. So again, they have a responsibility to entertain any offer on behalf of investors. So I think it's all fair game. I guess, just one follow-up, Josh. So like, what would be your advice to, the panelists here or an academic sitting in the audience, if they, if they wanted to really check for, real interest in their technology within a big pharma company, what would you say is the key to making sure that whatever that interaction is will give a person quality feedback? Is there a way to get that, or do you think it's a little bit of a hit or miss?
In a large pharma environment?
Yeah. And then let's do large pharma and then go down to, how a biotech might be different.
It's important to have a champion in R&D who has influence in the company and can drive to a deal. And I think it's always best to have R&D level champions. I mean, there are other sources of capital in large pharmas, like a lot of them have some kind of sort of internal venture fund where they'll make strategic passive investments that sort of without real buy-in from R&D sometimes, or like token buy-in. So I think those are usually the two parties that most small companies interact with, either the tech, the VC group within the company or the R&D. And I think if you're in a series of meetings where the seniority of the attendees is not going up or staying the same, you're not seeing things in writing, you don't feel like you're on a timeline, like the diligence field goalpost keeps shifting around on like what the next steps are. I think those are all subtle clues that it's kind of going sideways. And then on the other hand, if you're really seeing like a focused effort to get something done and you're seeing consistency of voice not just in your conversation, but with Yale Tech Transfer, that's an encouraging sign.
Great, thanks. So, so we're going to shift to some questions from the audience. Again, just to remind the audience that, again, we've disabled the chat room. So if you want to ask a question, though, raise your hand, and then the people supporting the logistics will engage with you and we'll get your question answered. So I think Guillermo Glazer has a question. Kathy, if you can open the mic.
Unmute.
Okay. Go ahead, your mic's open, I think. No, not yet. Guillermo, it looks like your mic still may be muted. Okay, try now.
Thank you very much. I would like to know whether there are guidelines. I am like an investor just looking around. If there are guidelines as to how do the pitch people pitch to investors to make sure that all the basic Yale ethical considerations are in place. In other words, how do we know that the information they are giving is 100% science-based and not tinged by the interest in getting funding, to be very blunt.
Great. Yeah, thanks for the question. I'm thinking, John Pusis, are you around? Maybe you can speak a little bit about sort of the venues used for these purposes, as well as sort of the curation of what goes in.
Sure. Thanks, Tim. So Guillermo, thanks for your question. When we have faculty pitching to investors or to companies, they're generally working very closely with with OCR, with my colleagues here in the Tech Transfer Office. So for example, I had mentioned earlier in the chat that there's a pitch fest being held in early December. These are the applicants for our Blavatnik Fund for Innovation Awards. So these faculty, I think there's about 30 of them this year, have all applied for this award. They're giving 5-minute pitches, and they're coached extensively, not only by people in the Tech Transfer Office, but also by a board of reviewers, which includes investors and also people from corporate VC funds, as well as people in the pharma and biotech community, our entrepreneur in residence network. And, you know, I think we all expect that Yale faculty are going to adhere to the highest standards of scientific integrity. When there's any kind of conflict of interest management plan in place, faculty are expected to make those financial interests, you know, known publicly when in publications or presentations or the like. So I hope that answers your question.
Thanks, John. I think there's been a couple mentions of the Blavatnik program. I think Ranjit had it in his slides. I think some of the panelists have been involved in this. I think, John, you just mentioned it. Maybe, John, I don't know if you'd be best, or even Ranjit, just to speak a little bit about that program, because again, I think it's one of the key parts of the infrastructure here, trying to help
Sure.
bridge academia into commercialization.
Yeah, I can give a little bit of an overview. Yale's been very lucky, and we're very grateful to have a generous award from the Blavatnik Family Foundation. This is an award or foundation set up by Len Blavatnik, and similar awards have been made to Harvard and to Columbia. So Yale's received a total of, I think, $25 million from the Blavatnik Family Foundation. And this is used to fund projects at Yale that have significant potential to have an impact on human health and medicine. And every year we have a contest amongst faculty at Yale who apply for these awards. They're between $100,000 and $300,000. And generally these are quite competitive. I would say they're significantly more competitive than an NIHR1 award, for example. And some of these have already advanced to the point of having significant venture capital investment. So for example, Citi had launched a company last year called Evolve Immune, which raised, I think, something like $35 or $36 million in an A round. And this was based on research that was in part funded by the Blavatnik Award. And Ranjit, I know you've applied for the award this year. You could, talk perhaps a little bit about your experience, or Sidi, talk about your experience in going through the process, perhaps.
Sure, happy to. I don't know, Sidi, if you want to go first, or so I'm not dominating the discussion.
Oh, sure. I think the Blavatnik process is extremely rigorous. We went through multiple rounds of training and multiple rounds of discussion internally first, fully validate the science, and then extensive discussion with the other partners. And we are fortunate to be funded based on the science by a panel of extensive committee from multiple entities, including academic, industry, and venture. And then we're able to advance the science because of the award, and then eventually able to create a startup. So, Ranjit?
Yeah. I'll just say, I've absolutely loved the process. We started a company called Therapeutics a few years back, and that was not through this process. And I was able to sort of compare and contrast. And the process for Athena, which was recently awarded a Blavatnik grant, and we're in the middle of the award. I can tell you it was a phenomenal learning experience. Each round of the pitches, you not only learn how to refine your pitch, but also from the VC folks on some of these calls would sort of give a lot of real-time feedback. I kind of think of it as a rapid acting SBIR that's internal to the Yale community. So I think it's a great opportunity. It allows you to do a little bit of de-risking still inside the walls of Yale before that, startup really takes off into the air. So yeah, really a big fan.
And I'll just add from being part of some of those venues, for those in the audience, maybe not as familiar with the ecosystem here. There are multiple venues, whether it's Blavatnik, pitch, and even precursors of those, where vetting goes on by people who've been there before, including scientists, venture capital people, who really pressure test opportunities. So again, I think there's been a nice build in an infrastructure around Yale, really to support the the growth and mentoring of scientists interested in translating their research to commercialization, and the quality is high because of those interactions. I do have a couple of questions from the audience that were questions that came in advance. Let me put the first one out there, and maybe Ranjit and Josh, just thinking about the nature of this question, it might be addressed by you. And the question is, are rare tumors disadvantaged, in sort of this translation from science to, commercialization? And this is someone who's interested in PNETs, primitive neuroectodermal tumors. So and Ranjit, I know you have some interest in these areas, and Josh, I know you've had certain experience in what might have been considered, rare cancers. Maybe if you two guys can just address that.
I'll have Josh start, because I've learned so much from Josh in my time that I've known him.
So the first drug I developed at Loxo Oncology was a TRK inhibitor for a population that in the United States was probably about 2,500 patients a year, assuming you could find every last one, which you certainly couldn't because of the state of the diagnostic testing environment. So candidly, I think big pharma is not seeking out those opportunities until there's an approved product. And then there's just so little in the cupboards of approved products that they can add to their commercial pipeline. They get interested after approval, when they can begin to think about pricing and think about, time on drug and prevalence modeling, and maybe they dare to dream once it's FDA approved, but they'll almost never green light. And I'm being really reductive here, and I'm not talking about my own company, which now I have some influence on their thinking, but I think most want to see that the commercial guys nip that in the bud at a lot of large companies early, let's just say. I think in contrast, Wall Street gives a lot of value to the rare company. The Wall Street places a premium on likelihood of success. And that's, I think, why rare tumors are exciting in that presumably they're mechanistically better understood, maybe more homogeneous in their biologic drivers, and therefore druggable with a higher probability of success. And so I always probabilize success over, I always value PTS, probability technical success, over commercial potential. And I'm weird that way. But I think Wall Street gets it. They just want to see great data. They want to understand, is this a company that's going to be worth greater than zero on their leader programs or not? And being in a group of programs that is on the way to an FDA approval or not is a big fork in the road for the biotech investing universe out there, and small companies. So like there's companies out there. There's a company out there developing a menin inhibitor for mixed lineage leukemia. There's 2 companies out there doing it, and they're able to attract capital and a following, and people are excited about being exposed to that. That's not a kind of program you'll run across in most large pharmas, though.
Yeah, and I'll add, as a sort of on the clinical side, there's really now an infrastructure that's allowing a lot of these rare tumor trials to progress much faster. As a adult and pediatric brain tumor doctor, we've been working on things like IDH mutant gliomas in kids, which is actually not that common. And we now have pretty robust cooperative groups that are ready to jump on sort of the precision medicine angle. And another interesting thing is a lot of these foundations are becoming more active. The National Brain Tumor Society, for instance, is started a venture fund to try to get the more rare tumors funded. Kidney cancer, we look at FH mutant HLRCC. This is like 300 cases a year, but there's actually now a whole group that's trying to fund these smaller trials. And then when you throw that together with the orphan status, priority review vouchers, depending on which area you're looking at, there are pieces of the puzzle that are coming together that are making this a little bit more tractable.
I think the existential risk of everything we just said is the drug pricing debate.
Yeah.
So if you say, if we can't accept that there's such a thing as a $20,000 per month or $30,000 per month therapy, then, and we create legislation or mandates around price caps and limits, I don't know how the financial model works, even at the small company level. And I'm not seeing enough nuance in the public policy debate that's really tackling that question.
Yeah, so look, and I would just add, so small tumors are great if you really understand the biology, because the therapies can have a tremendous impact. I mean, Josh's response rates with your products were in the range of 50, 60, 70%, just to remind us. 80%. I mean, so 80%. I mean, that's not only unheard of in cancer, that's unheard of in almost any indication across the industry. So when you have something so profoundly impactful, just because of the transformative benefit that has to a patient, that really is ascribed value. So, a small population with a really potent therapy, rightfully, can be valued highly, and therefore certainly draw investment, as Josh pointed out, from investors and from the small biotech community.
And I will add that there's a way to have a strategy where your first solid tumor phase 1 might have all comers, and you have a dream that your follow-on trial is for that population. Obviously, that's always difficult to walk that line, but if there is a way, certainly many companies have done it, then you can kind of hedge your bets a little bit. But
it still takes an enormous amount of capital to develop a rare tumor drug.
Yeah.
Our second drug was targeting 2% of the lung cancer community with RET alterations and medullary thyroid cancer. We were running GMP manufacturing campaigns that cost $30-40 million, just the manufacturing. And then you throw on top of that, it takes hundreds of millions of dollars to get a drug approved, no matter what the market potential is. It's just, there's these fixed costs of, compliance, manufacturing, preclinical, data management, post-safety, post-marketing safety monitoring. Like there's just the small product and the big product are exposed to the same fixed cost, issues. And so don't assume you can skunk work it forever. If the drug starts to work, you're going to need real capital and a real management team behind it.
Yeah.
So again, open up to the audience for any questions, raise your hand and we'll get you into the meeting. And Kathy, you make me aware of any of those that I'm missing. Well, let me just make an observation and then, pass on a question to the panel. So, one thing that I'm struck by about Yale compared to other institutions with worldwide renown is that while, there's an incredible depth of talent and science, around every corner, the institution has a smallness to it in terms of being able to interact with it, which to me is like really unique compared to other places I go. And I think that's a wonderful attribute. I see that also in the collaborations within Yale. So again, I know many of you, and I see a lot of collaborations going on, within the walls of Yale and within the Cancer Center. Ranjit, I think you called some of this out, sort of with the lab to the clinic. Maybe if you could speak a little bit more about that. And sort of my angle is, translation is so important and markers of translation are so important. And Ranjit, you talked about this iteration of lab to the clinic and the importance of tissue, human tissue tested with your drug and interrogating that to understand what's going on. So maybe Ranjit, I'll throw it to you, but I really would ask others really just to talk a little bit about some of their collaborations within the walls of Yale that they think are really unique and really have enabled you to do what you want to do.
Yeah, it's a great question. And as I mentioned, I've been at Yale a while. I was a resident at Sloan Kettering for about 5 years and was able to compare experiences at much larger sort of complexes. And one of the reasons I came back to Yale was that spirit at the Cancer Center and on the campus. It's a small, close-knit community, but with very, very big ideas and deep collaborations. And certainly Dr. Fuchs has really engendered that further at the center. And it's just a wonderful place to be. And just an example is, when we found that, IDH mutations, cause PARP inhibitor sensitivity, and it was sort of, the opposite of the current approach in the clinic to use IDH inhibitors that suppress the mutation. We would actually say we should exploit the defect. We still need to figure out if it's got legs in the clinic, and that's what we're doing. But it was the folks like Pat LaRusso and Paul Eder who run phase 1 here at Yale. And, we brought, literally, she's down the hall. Right when we're getting the paper ready, brought the galley proofs over to her. And I said, well, what do you think? Can we take olaparib and just run a trial and just get this going? Her and Paul Eder, we sat down, had a cup of coffee, and we mapped it out. And part of the reason it was so successful was that it isn't an assembly line type of approach. And it gets back to Josh's comments of at the upper level, if those people know the deeper science, same thing here. If the clinical trialists have a deep understanding and respect and relationship with the science, you can have conversations with pure basic scientists, anyone, and really integrate that data. And so when Pat and Jeff and I and Peter Glazer and folks are mapping those trials out, we're thinking about the on-treatment biopsies, the PK/PD biomarkers, and we're getting deep in the science. And, Pat has a deep respect for it, and we kind of go back and forth, and no one's making assumptions about, the other part of the coin, the trial design versus the science. And so I'll leave it at that, but that's just one example of why I think things have flourished here.
I also just add my experience with the Yale Cancer Center. I'm actually on the main campus, so I teach undergraduates biology. And so for me, the Yale Cancer Center has been instrumental in making sure that I know what's going on from a clinical point of view. The retreats, the seminar series, the pilot grants that are available to faculty to facilitate collaborations among people from different disciplines really make for a very tightly dense collaborative network. But I just want to put this two finer points on this with respect to my two recent companies. When I had this a bit of a crazy idea of how to degrade proteins, and I was thinking about how to start this company, Arvinas. I knew that I needed to have a killer app, right? And so the beauty of the platform technology, it could be applied to many different targets. And so I sat down with Dan Petrolak, a prostate cancer specialist here, and with my investor. And it was really nice to be able to sit and have the investor listen to the end user, if you will, hear what could be a unique application of this technology. And I think that really helped me seal the deal in terms of getting Arvinas off the ground. More recently, another platform technology company, we were thinking about indications. I was able to go to the leadership of the cancer center and say, listen, would it be possible for me to get together 3 or 4 basic researchers in cancer in this area, and just to have a sit down where we could throw out ideas and really whiteboard things, and really allowed for this nascent company to be able to have, again, real world interactions. And so it makes for, I think, a very unique place here at Yale.
Faye or Sidi, any comments, in regard to, some of your collaborative experiences?
Sure, I think Yale Cancer Center is a wonderful place, and it has so many different experts, and collectively we basically cover the entire cancer expertise in this place. And you can basically find the clinicians and basic scientists and physician scientists across the board, no matter what cancer type you're trying to hone in or to discuss with. And it's really, truly collaborative environment over here.
And Faye, anything to add?
So I would second all that has been said before. And I think one of the things that we've been primarily interested in is we know that our strategy can be used to treat drug resistance, and the opportunity to get patient samples so that we can make patient-derived xenograft models has been really instrumental for us to kind of see whether or not we have functional activity in drug-resistant tumors.
Yeah, great. Thanks for that, Faye. So we do have a question from the audience. I think Alexander. Let's see if we can get you activated.
Yeah, hi. Can anyone hear me?
Yeah, we can hear you.
Okay, thank you. Thank you everybody for coming together. It's been really, really exciting. So my question is open to anyone on the panel. I'm just curious about the broad cultural differences between industry and academia. Specifically, do you ever have or see very successful academic scientists and clinicians who, for personality reasons, experience a lot of friction in their involvement with industry? And conversely, what kinds of personality traits do you think predict maybe a smoother transition from academia to industry?
We'll open that up.
I love the question. Thank you for it. I think about it all the time. I think one of the biggest differences is the mindset around risk taking. In academia, it's okay to like place a lot of bets and be wrong most of the time, learn some interesting things along the way, get some publications even if you're wrong. I'm referring to, even like clinical publications, posters, etc. And in industry, you're accumulating a track record always. The projects that you launch and work on have to work. You need to see patient results, and you can't just take flyers all the time. It's kind of like being a movie producer in Hollywood. Like the last movie you make, if nobody comes to the box office, you don't get to make another movie. I'm not saying I don't think it's as reductive as that quite. I mean, we have a high failure rate in this business, but if you're just somebody who places bets because it's someone else's money, and like, there's a very common academic behavior of like, it's not my money. I'll learn some science. It's an option bet if it goes well on the upside. If it's on the downside, who cares? I'll move on to the next thing. Like that's a very, I'll just say that's a more academic mindset. And the more industry mindset is like, no, this, I'm bringing all my best relationships and credibility to bear on this one project. Better work in patients, better ultimately, hopefully become an approved product that turns into a marketed medicine for patients. Like that shift in mind is like very stark. And I see a lot of folks who come in industry sort of mid and late career from academia, and they think that they'll come in, be sort of an on-high voice of logic and science and reason, the resident expert, and be sort of off the hook on the harder parts of being right and on the harder parts of actually executing against the plan. And those folks don't do as well either. So I think both on like being right about your convictions as well as being a contributor to the execution against the plan. Those are the two behaviors that define success in industry, where in academia, there needs to be room for failure. There needs to be room for exploration and multiple ideas and moving on to the next project to keep the academic mission moving forward. And I'm not judging one or the other, but they're not interchangeable in terms of behavior.
Thanks, Josh. Anyone from the panel want to make a comment?
Yeah, I'll just add, because I know we're running out of time, but you know, in recent years, I feel like I'm learning sometimes the friction, because I think we've seen it. I personally believe it derives from sort of maybe insecurity on what we think we know and what we don't know. And this is really everyone involved in the drug development. And I think a very important principle is that from the process of just the molecule to getting it into a patient, to then caring for that patient for several years afterwards, every step of that process is enormously complex. And I thought I knew a lot as an MD, PhD, work in the lab and in the clinic. And with Cybrexa going into the clinic this spring, you know, unbelievably newfound respect for how difficult it is to get a drug just into the IND and into a clinical trial. So I think really understanding and mutual respect often will stave off some of that friction, in my opinion.
Thanks, Ranjit. Okay, so we are running up against the clock here. So again, you know, thanks for the conversation. It's been great. Sorry about some of the disruptions, but you know, hopefully, you know, we've been able to gather most questions. What I'd like to do right now is just turn the microphone over to Charlie Fuchs again, just for a few closing remarks from him.
Charlie, please unmute.
Sorry. I want to thank Tim for running a phenomenal panel and all the panelists for great discussion. I think it was really instructive in terms of the science and frankly, the challenges of what I think is at the forefront of cancer therapy. I also want to apologize to all the panelists and the audience on the disruptions, and I can assure you that we are going to make sure that we don't experience a similar disruption again. I hope you all will use this as an opportunity to advance what has been a great conversation, because as I say, we want to build partnerships. I think you've heard today about the challenges of those partnerships, and we want to make them smooth and successful, because ultimately, to really advance cancer therapy, it's going to require a community. So please feel free to reach out to me, to the panelists. We hope this is the beginning of a conversation. In addition, I hope you'll all join us again on Wednesday, December 9th at 1 o'clock, when our third in the series of Yale Engage Cancer will be defining mechanisms and biomarkers of sensitivity and resistance to anti-cancer treatments. And then finally, I just want to once again thank Tim. Tim Shannon is a member of the board of our cancer center and has been really a great supporter of all that we do, and including running today's session. And Tim, I'll turn it over to you for the final closing remarks.
Thanks, Charlie. Thanks very much. Again, you know, so just let me extend thanks to the audience, to our faculty panelists. Special thanks to Josh for carving out time of his day. Josh, we hope to see you more and more here around Yale. Again, all I can say is, you know, like most people on the call, this is my passion of trying to do hard things and, you know, getting them into humans and hopefully making a difference in humans' lives. So, you know, it's a great community to be part of. You know, Yale's got great things going on. It is an easy place to engage with. So, you know, in reflection of the name of the event, I would encourage all of you, again, who are interested to engage with Yale and the Yale Cancer Center, you know, to try to do some important things. So thanks, everyone. And with that, we'll sign off.